Thank you, everyone, for joining us tonight for a team talk on managing Microstegium. And we all know we need a team. We need a huge team to deal with this plant. Our first speaker is Luke Flory. He's a professor and associate chair at the University of Florida Institute for Food and Ag Sciences Agronomy Department. He's also a Fulbright U.S. scholar. His research focuses on the mechanisms and impacts of non-native plant invasions in order to understand long-term consequences of interactions between invasive species and other potential global change drivers like climate change, fire, and emerging pathogens. He has a lab, the Flory Lab, that explores both basic and applied questions in systems such as the highlands of Galápagos, coffee agroecosystems in Costa Rica, eastern deciduous forest in the US, and pine forest and managed systems in Florida. His talk is How Invader Removal Method Affects Native Plant Response. Our second speaker, Art Gover, his talk is called Trying to Put the Bee Back in Best Management Practices. A lot of us in the government go by BMPs. It's an acronym that we're more familiar with than most of the public probably. Art worked for 35 years at Penn State as a vegetation management researcher, and he worked with row crops, roadside and utility, turfgrass, and natural area settings. He also worked with the Mid-Atlantic Invasive Plant Council for a while. He was president and did a great job. I'll just throw that in. He worked for 15 years with the Pennsylvania State Park System to develop a statewide planning template and park level plans and implementation of the plans. He started, because he had so much extra time, like 5 minutes, back in 2008, he started a part-time commercial operation specializing in invasive species planning and management. And he does this since he retired, which I forget, it's been maybe 5 years. He's going to talk about his research and experiences of actually managing stiltgrass and make some recommendations for us. So thank you both for coming. We really appreciate it. And as soon as they're finished, we'll open it up for questions. So I guess take it away, Luke, and thank you again for presenting to us.
I was the one that suggested we do a team talk because Jill initially asked me, and I said, well, it's been a long time since I've worked on Microstegium management, and maybe some things have changed. When I moved to University of Florida about 12.5 years ago, a lot of my research shifted to the southeast into other parts of the US. Previously, I'd done a lot of research on Microstegium, primarily on ecology and impacts, just a little bit on management. So I'll talk generally about Microstegium for those of you that are not familiar. Unfortunately, I'm guessing many of you are quite familiar with it, at least have plenty of it and seen plenty of it. And then I'll talk some about the management work I did that's quite old now, I guess. I did the research about 15 years ago or so. But I think it's probably still relevant, but that's why I'm curious to hear what Art has to say. Okay, so Microstegium viminum, or stiltgrass, I don't call it Japanese stiltgrass because it's actually native to much of the southeast US, a whole lot more than Japan. Actually, the majority of its native range distribution is in China and other parts of the southeast US, so I just call it stiltgrass or Microstegium. It's an unusual species in that it's a C4, meaning it's a warm season annual grass that is highly shade tolerant. We have a paper showing experimental shade at about 7%, and there's still reproduction. And so it's pretty incredible in terms of its shade tolerance, which means that it may fill this empty niche in eastern deciduous forest where we do not have many shade tolerant annual native species. And so it's doing most of its growing at a time when there aren't many native competitors. It has very few herbivores or pathogens in the introduced range until the emergence of these foliar fungal pathogens that I'll talk about at the end of my presentation here. It now occurs in, I don't know, I haven't looked lately, 26, 27, 28 states. This EDMAP's county level distribution map is a bit dated, maybe a year or two old, so it's probably more widespread than this. The GBIF map at the bottom left shows that it's throughout the eastern U.S. as an invader and native in other parts of the U.S. or in other parts of the world, primarily Southeast Asia. It occurs in a wide variety of habitats. When I first started working on it in 2003, 2004, with funding from the Nature Conservancy in Indiana, some people said, oh, it's not much to worry about. I only ever see it along roads and trails. Well, just like many invaders, you first see it in disturbed areas, but then it can invade a really wide range of habitats, from shaded, undisturbed forests to full sun, and from full sun to less than 5% light. I've seen it at Big Oaks National Wildlife Refuge, which is a former military testing facility in southern Indiana, which is highly restricted in terms of access, and I've seen it in areas that humans probably haven't been in decades. So it does not require human disturbance to establish. It can create extensive invasions. So here are some photos. Top left is in Virginia. The one on the right is in Indiana. You can see the small seedlings on the bottom right. It's super prolific annual species, which I'll mention again when I describe the models where we've been trying to figure out what the implications of this emerging pathogen are. And it has all sorts of impacts. And so I spent a lot of my PhD dissertation work, time during my PhD, working on my dissertation on this field experiment you see on the bottom left, a number of other experiments that I also did during postdoc work on fire invasion interactions. We published a whole suite of papers on how it impacts native biodiversity, tree regeneration, arthropod abundance and diversity. In total, I think I've published almost 25 papers related to Microstegium. But what I want to talk to you about now, that I think a lot of you are probably most interested in, is what do you do about it once you have a major infestation? So I started this experiment at 8 sites in southern Indiana in 2007, I believe, 2006. And we had 4 types of plots that we set up at each of the sites. So we had plots that were randomly assigned as controls, plots where we carefully, as we could, myself and a group of undergraduates, hand weeded Microstegium while trying to minimally disturb the native species. One where we applied a post-emergent grass-specific herbicide. In this case, it was Fusilade, and there's a number of other post-emergent grass-specific herbicides that are supposedly similarly effective on Microstegium. You can actually spray it in quite low concentrations, and it's still quite effective. On an individual basis, Microstegium is very, very wimpy. There's just a lot of it. And then our 4th treatment was that same post-emergent grass-specific herbicide plus a pre-emergent herbicide. Pre-emergent herbicides, at least as this one in this case, and I don't know of any that are any different, they kill seeds as they germinate before they emerge. So they're pre-emergent. They don't actually kill the seed. They kill the kill the plant as it germinates before it emerges out of the soil. So we applied each of these treatments randomly to a whole bunch of plots and monitored what happened over the next couple of years. So in terms of treatment effectiveness for Microstegium biomass, when we measured it in the fall of the year that we did the treatment, we see very little difference. So hand weeding was not particularly effective for some reason in the second year. In the first year, it was quite effective, but less effective than the herbicide treatments. So the different letters over the bars indicate significant differences. Both the post-emergent grass-specific herbicide and that post-emergent herbicide plus the pre-emergent were equally effective. So like 99% plus Microstegium biomass, less biomass than in the reference or the control plots. However, it's a very different situation the following spring. So Microstegium cover in the spring after those 2 treatments was quite high, about 75% as much cover with the hand weeding as in the control plots. And that's probably because despite our best efforts, we missed a few plants. Those plants went to seed, or there was seed in the seed bank that emerged, and it emerged at especially high numbers because of the disturbance due to the hand weeding. The post-emergent grass-specific herbicide was much more effective, especially by the second year, when there was more than 95% less Microstegium cover with that post-emergent grass-specific herbicide. Now, when we added that pre-emergent herbicide, there was almost no Microstegium. It was virtually undetectable, and that's because this pre-emergent herbicide is extremely effective at preventing individuals from emerging. But what about the native species? This is the part that is, I think, the most interesting and important here. So in terms of native species biomass overall, so this includes individuals that lived after our treatments and grew larger, or emerged, established from seed after the treatments. There was more biomass in all of the treated plots than in the reference plots. And we're not sure exactly why, but they were released from competition with Microstegium because it was removed. And they were all, equally successful in terms of reestablishing native biomass. Diversity was a different story. So we got higher diversity levels in the hand-weeded plots and the post-emergent grass-specific herbicide plots than when we added the pre-emergent herbicide. Now, of course, that's because the pre-emergent herbicide kills all species as the seed germinates. So diversity did not increase under that treatment. I think one of the most important findings of this study is that tree seedling establishment was a lot higher with the grass-specific herbicide, and we did not see that rebound in any of the other treatments compared to the reference plots. And so there was no more tree seedlings in the hand-weeded and the post-emergent plus pre-emergent treated plots than in the control plots where Microstegium was left intact. So this was the major finding that was super important from this study. And you can see the result in the picture on the left. I've added the dotted line to show the plot outline. You can see the Microstegium all around, and you can see the native species returning into that area. These are small plots. They were only 2 by 2 meters, but we had 320 of them. That's why they're so small. So the paper that came out of this was called Invasive Plant Removal Method Determines Native Plant Community Responses. And it was important and continues to be important, I think, for this species, invader, and for others, because it shows that we need to really think about how we're getting rid of invaders. I'll show you on the next slide a table where I emphasize there is no reason to use glyphosate, especially since glyphosate is now more expensive than it used to be, and pre-emergent or post-emergent grass-specific herbicide prices have come down. There's no reason to use glyphosate on Microstegium ever, in my opinion. So hand weeding and herbicides were effective at removing Microstegium, but only the herbicides provided long-term effectiveness. The pre-emergent herbicide was super, super effective, but it inhibited recovery of native diversity and tree seedling establishment. And so the recovery of different plant functional groups depend on method of removal. So here's this table that I mentioned. This is one that is, I'd like to hear what Art thinks about it. This is one that I put together when we were putting out some extension publications through Purdue University when I was still in Indiana. So we did a lot of work with fire, and I would not recommend it. We need more information to see about properly timed fires and if it can help control invasions. We have a paper showing fire invasion interactions are really bad for certain tree species. Again, no reason to use glyphosate. Super effective at getting rid of Microstegium, supposedly, but you don't get anything else back either, obviously. Grass-specific herbicides can be used. Highly effective, allows for return of native diversity and tree seedling regeneration. Hand weeding's fine for really small infestations, but it's impractical for large invasions, which if you have some Microstegium, you probably have a lot of Microstegium. Mowing is possible. I participated in a study where we tried some mowing. Obviously, it's not specific to certain species. It's fairly indiscriminate. You can do some string trimming. I've heard of people having success with that, but even if you're a really good string trimmer, it's hard to be very very good at avoiding the native species. And then pre-emergent herbicides, I suppose you could use them if you were confident that almost everything you had was Microstegium and you weren't trying to establish any more native species or trees or something. But grass-specific herbicides, I think, are really where it's at. One thing I'll mention here is I am not a herbicide person. I'm not a weed scientist, not trained in it. Definitely check with your extension folks about any kind of concentrations, what products to use, where to use them, check the label, all that stuff. Just a disclaimer there. I will mention here, we just published a paper in Restoration Ecology on Imperata cylindrica, which is Kogan grass. It's a massive problem in the Southeast US. And we found very similar results where if you use a grass-specific post-emergent herbicide, you can get effective control of this other grass invader, Imperata, and have native species come back. It takes a few more applications, but it can be effective, and I would argue is worth the extra cost if it does take you a few extra applications. Okay, so then the other thing I want to talk about briefly, because I'm guessing many of you have seen this. This is what we started to see, I don't know, about 7 years after I started working on Microstegium, 6 or 7 years. We started to see a lot of populations with this emerging pathogen, this foliar fungal pathogen that we eventually identified as a bipolaris species. We eventually called it Bipolaris megaspora. We've now formally described it with USDA. And when we saw this, we thought, hey, well, you know, invaders, they escape these natural enemies. It makes sense that things would catch up with them. And my former advisor and I published this paper showing this expectation, or possibility at least, that as an invader increases in spatial distribution and density over time, that pathogens or other enemies would catch up with that invader and possibly drive it into decline. In this paper, we had some photos and examples of other invaders that have emerging pathogens documented on them. So with my first big federal grant, once I got to the University of Florida, we went out and surveyed more than 80 sites in 18 states and identified a bunch of pathogens, primarily Bipolaris and primarily Bipolaris megaspora. Now, interestingly, one of the primary predictors of occurrence of Bipolaris was the age of the population, suggesting that this pathogen was accumulating over time. The other thing that we did in West Virginia at this experimental forest was to apply fungicide on Microstegium populations that were infected. And what we found is that at these different sites where we applied this fungicide, after the second year, there was about 90% more seed production, suggesting that the pathogen was impacting reproduction of Microstegium. And that's where that study ended. So we published the paper in Ecology Letters in 2016 and said, there's a lot of this emerging pathogen. It occurs more on older populations, and it affects reproduction of Microstegium. We then got another grant and added a bunch of team members that were theoretical ecologists and plant pathologists and other field ecologists to test the idea of what happens with interactions with native species. So one of the ideas is that the invader could go into decline, native species could return. We called this the PAID hypothesis, pathogen accumulation and invasive decline. We also thought, though, that there could be no effect, that it appears to have an effect on reproduction or has some effect, but it doesn't affect it at the population level. Then the third possibility is that the pathogen could accumulate on the invader and spill back onto the native, disproportionately affect native species, and therefore release the invader from competition and make the problem even worse. And so I'm not going to show you this research. It occurred over about a 3-year period, and we're still working on the data. But what we did was to look at all the interactions in terms of competition and pathogen transmission through the litter and through live leaves, and interference competition, meaning the accumulation of all that dead thatch impacting the invader and the native species. And we set up this whole series of experiments at Big Oaks National Wildlife Refuge in Indiana. We published a whole series of papers about what was happening with the fungus and the litter and plant competition. And then the last thing I'll show you is that we put this all together in a model, and I'm showing you our current model results. This is not published yet. But what we're seeing here is relative biomass or disease severity of the invader and the native competitor. In this case, we are using an Elemus species, a native perennial grass species. Prior to invasion, you see high abundance of the competitor and no disease. Once the invasion occurs, in our model, and this is based on all data from greenhouse and field experiments, in our model, the competitor declines a little bit, but not too much. And the invader, Microstegium, goes into high abundance, of course. The dynamics change dramatically when disease is introduced into this model. You see 18% lower biomass, so there is an impact on the invader. The disease definitely affects Microstegium, but there is an 88% lower biomass of the competitor. And this is because the pathogen spills back. It accumulates on the invader, which creates huge amounts of spore production. Those spores then spill back onto the competitor and reduces its biomass by almost 90%. So despite the fact that people see Microstegium apparently dying out, our model suggests that is unlikely to occur. And anecdotal field observations suggest that's true. There's a site on what's called J Road in Big Oaks where I first saw the pathogen, I don't know, 15 years ago. And every time we go back there, it's heavily infected and highly abundant. So despite the fact that people were very hopeful this pathogen was going to make Microstegium go away, and I have actually heard of people spreading the pathogen, it can actually make the problem worse.
Thank you, Luke.
Well, good evening, everybody. I'm Art Gover. I will continue our discussion of stiltgrass. Trying to focus on a little more applied elements. I do just want to take a moment to acknowledge that even though I am retired from Penn State now for almost 3 years, a lot of what I'm going to share with you are resources, images, information gathered while I was working there, particularly during my work with the state park system on the invasive species planning, prioritization, and implementation work. I want to share some resources with you. At Penn State, there's 2 websites in particular. There's the Wildland Weed Management site that does have a selection of end user documents. For instance, this one entitled Herbicide Selection and Use with a focus on natural areas. There is a series of species specific fact sheets. They're cryptic isn't the word. Anyway, you'll notice 2 pages, no pictures, anything like that. Kind of the premise is, you know what it is. Let's get right into prescriptive details to manage these species. Penn State Extension also has an ever-expanding series of not only fact sheets, identification videos, and so here's, for instance, their Japanese stiltgrass fact sheet. Much of the information from the other one, but now you do have the identification information as well, and high-quality images. Here's some review. So when we're talking about Japanese stiltgrass, I will just say stiltgrass. So it is a summer annual grass. Germinates spring, terminates its life before fall, before winter, I should say. Warm season grass, so that C4 metabolism, very shade tolerant. I'm just parroting what I think most of us read, first reported in 1919 in Tennessee. Someday, when we were blessed with the presence of Les Meerhoff, he would do some wonderful time sequences when he was describing the invasive plant atlas of New England, showing by way of herbarium records the advancement of species. And I would really love to go into, particularly now that a lot of them are digitized, but go into the herbarium records and really use that as a means to show the spread of stiltgrass. Now, in my lifetime, I first laid eyes on it in 1991 in southeastern Pennsylvania. For a few years, it was just a rumored thing. I did finally see it in Bucks County, but here where I live in central Pennsylvania, I started seeing it in the state forests along the unpaved roads probably 1995. Since then, it's everywhere. It's there's nothing novel about it. If there's any novelty anywhere with stiltgrass in Pennsylvania, it's that if you can't find it. This is the species as we've seen before, but characterized by that asymmetric leaf and that silvery smudge along where the mid vein would be, kind of a stand level picture. I've heard people describe it as like miniature bamboo. I'll just phrase differently what I think Luke pointed out very well. So it is at the individual plant level, it's easy to kill. As a species, because of that durable, persistent seed bank, very hard to manage. One way to look at it when we were doing, well, when we're still doing, when we prioritize management at a property level, we're looking at the ecological integrity, the 4 factors we're looking at: the ecological integrity of a site, the extent of a given invasive species. We do look at its impact, which is more of a bonus point, but one of the key elements is what we would call restoration effort, which we were scoring on a 0 to 3 scale. And if you look at this graphic here, where your y-axis is control effort, x-axis is time, the premise is you're always at kind of maximum effort when you start, but you're going to give this higher score to a species that, like number 3 here, the green line, that actually responds to your efforts. And let's say in a 2 to 3 season time span, transitions from being a control phase infestation to a maintenance level infestation. And one of the challenges with seed banked annuals is they tend to be somewhere more like that 0 line, where if the seed bank is there, you just keep seeing the same response from the species year after year. And I think I should stress, when they're established, I enjoyed the figures that Luke shared. And when I think back to some of the sites where we first started working, so early 2000s, we would see where we had successful suppression the first year of an investigation. You could see those plots the next year because they stood out pretty clearly in that checkerboard pattern. But returning to those sites later and doing similar work, we would see, you might still see complete suppression. But if we weren't using some sort of residual herbicide, the next season, it would just come back to the same level of stiltgrass. And on some of these older infestations, that's the phenomena we're accustomed to, and sort of caution people that that's really what you're dealing with, is all the elements that deposited that seed there are probably elements outside of your management control. So the first thing, usually when people say to me, let's just say as a professional operator, can you get rid of this stiltgrass? Usually I just say no. But what I can do is, if you are clear on what sort of plant community you want there, I can help you get to the point where you have your established plant community, providing those ecological benefits that you want, with a detectable population of stiltgrass there as well. And part of the issue, certainly where we are, is that interaction with deer. Commonly described as ecosystem engineers, and they're very common in Pennsylvania. And we complain all the time about our hunting regimen here compared to what you have down in Maryland or in Virginia. And kind of one data point to sort of illustrate that a little bit, this was some work done at Hood College by Kai Karaher. And I'm sorry, I forget which hurricane created all the blowdowns in the Catoctin State Forest, but they took advantage of these openings in the canopy to set up areas that were either ex-closed. You created these areas protecting sites from deer, or you would put up the posts but not the fence, so the deer had access to the site. So if you look at this particular chart where they looked at the beginning and then 3 years later, so your blue line are the exotic plants, the green line are the native plants, that where you had the exclosure preventing the deer from getting in there, you had a greater proportion of native plants after 3 years compared to where you did not have the deer. And just speaking anecdotally, they kind of described it. It was the plots behaved as if succession was occurring. Annuals like stiltgrass, like mile a minute, were prevalent almost immediately. But where you had the deer being excluded, they behaved like colonizing annuals and gave way to native woody species, and their presence became much lower. Whereas if you're not excluding deer, it's just not uncommon to see what appears to be just vast vistas of stiltgrass, kind of like this, where almost as far as you can see, a near monoculture of stiltgrass with maybe some white snake root popping out of it. Take some more looks at stiltgrass. So here's some of its phenology. It is, I would describe it as a very plastic plant. So at high density, high stem densities, there'll be hardly any branching. At low stem densities, it is quite an expansive and sprawling plant. And it will root at the nodes. So this example here, you can see the white things that look kind of like corn brace roots coming off the nodes of this plant. And, those are the stilts of stiltgrass. This is kind of what it looks like right now. This is kind of pick an unpaved forest road here in central Pennsylvania, but that's what we're looking at. Tremendous amounts of biomass accumulated from the previous season, just waiting for this year's growth to come up through there. Seedling shot, so the little, the coleoptiles are almost circular, very short, very wide. And we expect to see that almost any day now. Some approaches to managing seed banked annuals. One would be kind of a direct approach where you work very hard at preventing and reducing the seed bank. And I think what I would stress here, which I should have added, like this is just ongoing. If it's there, you go after it. But this would only be sustainable, arguably, on very small areas. So, when I'm talking with land managers, for instance, if you have a population of rare plants that needs protection, this might be the approach you're taking. But for larger expanses, usually what you're doing is, well, I'm going to refer to it as an adaptive approach, where the objective is identify the plant community you want, and that plant community might be tempered with the management efforts to get there. Plan on managing or suppressing stiltgrass for a very finite period of time, 2 to 4 years, where you release that desired community to the point where I'll say it's sustainable. It can withstand the presence of the stiltgrass, and you just kind of pat it on the head and you move on to your next project. So let's kind of get at some specifics here. We'll revisit integrated pest management. I'm sure you're all familiar with this. I hope you are. But let's review the fundamentals of integrated pest management. Is you start with a clear idea of what, in our case, what your plant community objective is, because without that, you don't know what your pests are. So with the objective clear, the pests identified based on the objective, you can set your thresholds, which determine when you're going to intervene, and then you unleash your tools. So cultural, mechanical, biological, and chemical techniques. A lot of cultural techniques, you could describe them as indirect. Phytosanitation or prevention is a big key to this. So, here's an example of a tracked mist blower in a forest setting, just full of Microstegium in its track. So just making sure that contaminated equipment, contaminated mulch, contaminated plant material isn't coming onto your site would be a key example of cultural management. Creating, maintaining, enhancing competitive ground covers would be another approach. Reforestation. So in this case, you've planted the trees and you're providing that control around the tree shelter to allow that tree to get established and I'm going to say released from the stiltgrass. I saw that the question about prescribed fire, and I don't know if prescribed fire is useful. Pennsylvania is increasing its use of prescribed fire. Unfortunately, I don't think the monitoring of the plant communities really is keeping up with it. Just in kind of, just a few instances here and there, I feel like prescribed fire just kind of plays into the hands of stiltgrass by removing any sort of biomass at the surface layer and heating the surface up a little more quickly. So far it's a non-zero, but close to zero number of data points, it doesn't seem to be a useful tool. You have pulling as a technique, fine for very small areas, not something you're going to implement on a larger basis. I will echo what Luke said about mowing. Late in the season has been my experience, and by late, I'm saying after July, cutting as low as possible. I do think string trimming is the approach where you essentially try to take it right to zero. There is biological control. This is just my stock picture for biological control. I'm not aware of any arthropods being contemplated for stiltgrass. This happens to be the mile-a-minute weevil, and Luke gave us that explanation of the fungal pathogen, but that's an example of classical biological control. And we'll transition into chemical control. This would be pre-emergence herbicides. You're applying herbicides prior to the emergence of the plant and controlling that seed, that plant as it's germinating from the seed. Post-emergence, probably no stiltgrass in this picture, but just showing you that it's after they've emerged and you're targeting emerged plants. I'll look at herbicides or kind of categorize them as, I'm going to say selective pre being pre-emergence. These are herbicides that have limited or almost no post-emergence activity. So if you apply them to an emerged seedling of stiltgrass, likely you would see no effect. This class, this grouping, this categorization of herbicides only affects annuals. These plants have to be coming from seed. A second category I would look at is what I'm going to loosely call pre-post. So these are material, they might be, so these are materials that have soil activity, but they also have some foliar activity as well. So that changes the timing that we can use them. And finally, post-emergence herbicides. So these are herbicides that are only useful on emerged foliage or targets. We'll focus here on the top, that summer annual line, because really the time frame that we're talking about working here when using chemicals for pre-emergent, early in the spring, before emergence or soon after. And with post-emergence, really the key there is, I should point out those columns, JFM ending with OND, those are your months of the year. So you could be doing post-emergence work as soon as April, though not likely. Probably more May would be the start point, at least in this part of the country, when the plants are big enough for you to detect and treat. Ending in August. Here, we tend to expect to see that inflorescence emerging from stiltgrass last week of August, first week of September. I don't personally know, haven't studied it enough to know when you have fertile seed, whether that cleistogamous or chasmogamous seed, but I think operationally it's just a good mindset to try to get your work done before you start seeing seed head emergence. So let's talk about operational calendars. So if we're doing pre-emergence applications for stiltgrass, you need to do it before it emerges. So that's why we're indicating February and March are your targets here for that material to be applied to the soil and to typically through rainfall to get to where it needs to be to affect that germinating seedling. Some examples of what I'm calling selective pre-emergence, and I'm making the distinction here again. These are pre-emergence herbicides that are limited to affecting annual species, hence the selectivity. I mean, there's certainly, industrial type herbicides that, you can kill trees with by applying to the soil. That's not what we're talking about here. These are materials typically you're going to see used in the landscape as well, and quite often in row crop agriculture. The way these columns are set up, you have the herbicide common name to the left in italics, a product example, and it's, for what we're talking about tonight, just a minor point, but the Weed Science Society of America does classify herbicides by their site of action, which from an herbicide resistance management point of view is a key piece of information. The common examples, pendimethalin or pendulum, prodiamine or proclips, rhizolan or surflan, and these are all WSSA group 3s, or what you might know as dinitroanilins. These are the yellow and orange materials, but they've been around a long time. There's a lot of use experience behind them, readily available, not that hard to use. A newer material is indaziflam or esplanade that has a different site of action. That's a group 29. These are useful where you have established perennial vegetation, because remember, they're going to be affecting the annual vegetation. So if you already have a perennial population that you're satisfied with, these could be a useful tool for you. And in forest, reforestation, or even meadow settings, if you have that diversity in the meadow that you want. They also come into play for vegetation-free situations like around tree shelters where you might be combining them with glyphosate. And just stressing once again, they are going to prevent the recruitment of pretty much the whole spectrum of annual species, desirable or undesirable. So here's an example. So here's a heavy stiltgrass infestation. This would be July in southeast Pennsylvania. So quite a mix of vegetation there, most of it Japanese stiltgrass. Similar, well, it's the same setting, same day, an area that was treated with pendimethalin 90 days before. So really this picture is predominantly goldenrod. So herbaceous perennials were not affected by the pendimethalin. The stiltgrass isn't present. So that's kind of the typical approach you're looking for. As we get into these pre/post materials, two ways to look at them. They could be standalone materials. You're applying them then at a full season rate, or this reduced selectivity. Well, because of that, you get the reduced selectivity because of that higher rate, or as a tank mix partner. And I should say a reduced rate tank mix partner. And what we're doing here is we're adding them to selective pre-emergence herbicides. So the ones we mentioned before, like pendimethalin or indaziflam, but at a very low rate, because what we're counting on there is they have foliar activity, and at very low rates, you can still benefit from that foliar activity, because what we're talking about is treating stiltgrass basically soon enough after emergence that you can detect it. So it's still very small. So the amount of foliar activity you have is enough to take out that emerged population of stiltgrass. The residual effect of that reduced rate is short. And what you're really counting on is for this material to largely selectively remove the emerged stiltgrass while you're waiting for that pre-emergence material, that selective pre-emergence material to get down into the zone where the germination is occurring and kind of carry you the rest of the season. So it's a means to allow you to, when you're treating areas where you know the infestation is, instead of broadcasting it, you can do the application where you see it. So it is one means of limiting the amount of material that you're applying. And common examples of herbicides that we might use for this would be sulfametron, commonly used as oust, imazapic or plateau, and then finally another example would be flumioxazin. Oust and plateau are group 2 herbicides. The flumioxazin, the SureGuard, is a 14, and as we'll see, that makes for some interesting use patterns. In general, if you're going to use these materials as a standalone, they are pre-emergent herbicides. So you could use them in that February-March window, but you can also use them later. And I think I would stress here, you're not using them to emphasize any selective element of them, particularly if you're using them later in the season. This would be kind of the mindset, for instance, if you're working in forest settings. And maybe you're coming out later in the season and using an application that not only is targeting Japanese stiltgrass, but for instance, hay-scented ferns and other ferns, as well as, I'll say, understory competing woody vegetation. Now, if you're looking at it as this low-rate tank mix partner, then you're partnering it with a selective pre-emergence herbicide. So you are using it early in the season and potentially extending that use maybe as late as May. So kind of back to the idea that, with that low rate tank mix approach with these pre-post herbicides, you could wait till this stage when you can visibly detect the stiltgrass and then apply that material instead of guessing based on residue where it's going to come up. Looking at them individually, sulfametron is primarily used in forestry settings for site prep and post-harvest applications, either anticipating disturbance and release of stiltgrass or right after disturbance when you're anticipating stiltgrass. Later in the season, it is combined with glyphosate. Pretty much at that juncture, in a forest setting, you're kind of going after everything. But this might be a typical application type for using sulfametron in a forest setting. So using a mist blower, trying to go at very low volumes per acre, like 5 gallons. So each sprayer load here might cover a full acre. If we're going to use oust as a standalone material, you're typically going to go, well, between 1 and 4. Label rate highest is 6. That's kind of excessive. If you're using between 2 and 4 ounces later in the season in a forest setting, you can typically anticipate also having residual control the following spring. So by waiting later in year 1, you do get stiltgrass suppression in year 2 with this approach. Stressing though, this is not a selective approach. This is, you're either releasing seedling trees that are there or creating an area where that's likely to happen. If you're using oust as a low rate tank mix partner, you're down at like 1/4 to 1/2 ounce per acre and adding it to these pre-emergent products like pendimethalin or indaziflam. And once again, that would be pre or very early post-emergence. Imazapic or plateau, it's kind of marketed as a soft residual for industrial settings, so you can use it where industrial settings are, at that margin with more ornamental landscapes. It's also very useful in native grass establishment. So big and little blue stem Indian grass, your gramias, side oats, and blue, and buffalo grass are all tolerant to imazapic, as well as the select forbs, and quite a number of those are legumes. So you can use, as I'll show in a second, it's if you want to maintain an herbaceous opening where you have stiltgrass pressure, using an amazopic tolerant seed mix is an approach that we use fairly commonly. Ernst Seed in Meadville, Pennsylvania has a specific plateau tolerant seed mix specifically for that. A potential drawback to using a mazopic is I can't say across the board, but from observation in the label, it is pretty active against your non-grass graminoids. So, if you want sedges, if you want rushes, this is not the material to use. I've seen very low rates pretty much eliminate species like soft rush from wet meadows, which was kind of counter to the idea. But here's an example of a site that was treated with plateau herbicide before the seed emerged. So everything you see here grew up through the plateau. So a lot of black-eyed Susan, there's partridge pea, there's cosmos in there as a cover crop, and there's native grasses coming in there as well. If we're looking at a mazopic as a standalone, you're going to be at 4 to 8 ounces per acre. And if you're using it in that low rate tank mix, I prefer 1 ounce, but I'm saying up to 2 ounces there. You're really trying to thread that needle and use just enough to kind of knock down any emerged stiltgrass, but not have effect on other desirable species in that mixture. And finally, we'll look at flumioxazin in this pre-post scenario. This has got a wide range of uses, nursery, landscape, agronomic, as well as industrial. It has contact activity, so you will see foliar activity when you use it, but it is short-lived. It's not a systemic. It's not, I shouldn't say it that way. It's not translocated throughout the plant. So particularly perennial vegetation that you treat with this, it's going to look horrible for about a week, and then it's going to start to grow out of it. And it also has aquatic labeling on a number of its products. So if you're looking for a product that, even if from a regulatory point of view, you're not in an aquatic setting, the fact that you're using an aquatic labeled herbicide might give you the option of, well, you're definitely going to be in a reduced risk situation with non-targets. An issue with flumioxazin is there's only a few products that allow you to use it selectively. So even though there's probably dozens of flumioxazin products out there, and I should stress except for the andaziflam, every product I've mentioned to you, it's off patent. So there are generic equivalents out there. And same thing with flumioxazin. So the product you would have to look for, if you're looking for it to use it selectively, where you're not trying to create bare ground, you need to use SureGuard SC or one of the generic equivalents to that, because a number of them are very explicit that they're to be used to create bare ground. But just a little, oops. Yeah, sorry. For standalone applications with flumioxazin, you'd be at 8 to 10 ounces per acre. When you're going to do that low rate tank mix, and I think I really like this product out of all of the three for this application because it's going to have the least spectrum. As I mentioned before, it might look the most dramatic because there is a considerable burn on a lot of species. But if they're perennial, they will regrow from that. So it gives you this option to come in later, let that stiltgrass emerge, kind of burn off what's there, and then release the rest of that herbaceous planting that you have. I'll transition finally to the post-emergence herbicides. And I see I've misspelled glyphosate here. So glyphosate, common example would be Roundup Custom. Sticking with aquatic labeled materials, you'd be looking at things like Aquaneat. Older examples would be Rodeo, which isn't made anymore, but you still might find it. Glufosinate is another post-emergence herbicide. You would see that as Finale. And then Luke mentioned the grass-specific herbicides, and there are a number of them. And if you look over there in that WSSA group in the far right column, these are group 1 herbicides. So they're active only on grasses, not on dicots, not on sedges, rushes, other monocots, very specific to Poaceae. Herbicide names then would be Quizalofop, Cethoxidym, Phenoxaprop, Clethodim. Luke mentioned his work was with Fluazepop. Or Fusilade. When you're looking at kind of their use, so glyphosate and glufosinate are non-selective. Everything you treat with them is going to be impacted. And with glyphosate, there's a good chance you're going to kill them. Glufosinate behaves a little more like a contact herbicide. So that annual species, it should be very effective against, but as you are treating perennials or really well-established annuals, there's a good chance there's going to be some unaffected growing point and they're going to regrow. The grass herbicides, it is really just grasses. As I mentioned before, that's all they're going to be active against. And I saw in the chat the question, are these safe to trees? Well, I should never use safe. Can you use them around trees and not injure the trees? And almost across the board, the answer to that would be yes. And if most of these, they might have ag labels as well as, let's say, landscape or non-crop labels. The landscape labels, many of these products have labeling specific for applications in landscape beds and around woody plants. So definitely low risk to young trees. Looking at the post window, really you're working in that kind of May to August window. Stressing again, we're trying to get done on stiltgrass before seed head emergent. Looking at glyphosate, non-selective, non-residual, so it's not leaving activity in the soil. You can use it to spot treat. And I want to enforce what Luke said. Everything that's in the patch you treat with, it's going to get taken out. So based on what your end result is, now, if you, like, for instance, there's a number of forestry projects where I'll get engaged. These would be NRCS equipped projects where the landowner is obligated to remove exotic shrubs and a lot of the herbaceous vegetation. And in that case, the stiltgrass just gets treated with the woody vegetation, so with a glyphosate-based mixture. And as we'll see in a minute, when you want to be selective, this isn't the approach you would take. You can do more broadcast type application where, you're taking out everything. So let's look at glyphosate. Let me back up a second, because one of the things, okay, I thought I was going to emphasize there, you can use glyphosate at a low rate because stiltgrass is so susceptible. So different site where we had done some research, this is what the control looked like in the middle of August. So I should point out that's a lot of stiltgrass in there. Treated even with only 6 ounces per acre of a glyphosate. So in this case, this was rodeo. Normally, like if you were treating a broader spectrum, you'd be at 48 to 96 ounces per acre. So, you're at a 1/16 to an 1/8 of a rate here and eliminating still almost everything. So even at very low rates, not selective. Glufosinate, it's non-selective, non-residual. It is less systemic than glyphosate. So you could potentially use this in situations where, well, if somehow there's utility in being non-selective, but having your perennials regrow. Let's put it that way. Really, if you want that selectivity, you're going to use a group 1 herbicide because they're only affecting grasses. And let's skip down to that idea of preserving desirable grasses. Stiltgrass is very susceptible to a number of these. I mean, everyone I've listed will definitely control stiltgrass. The question that you might want to get at is it really sensitive to one over the other? Because if it is, now you can start thinking about applying just enough of that material so that the stiltgrass is suppressed. You've kept all of your dicots. You've kept all of your non-grass graminoids. Can I keep other grasses? And our experience is definitely yes. I settled on using Assure 2, probably around 2002. We had done some comparative plots. This was not hard-hitting, weed science journal caliber research, but it was pretty apparent just visually that even though they were all active, the Assure 2 was more active. And you can use that at that 4-ounce rate. It's very effective against stiltgrass, but it'll leave other common native grasses that you'll see with stiltgrass, such as whitegrass or nimblewill, which, is in your lawn, you hate it. But, if it's in a natural setting, it is a charming native. So same thing we showed a minute ago. Here's the site in the middle of August, plenty of stiltgrass, just a few feet away in an Assure treated plot. Had been treated in June, so the stiltgrass would have been just a few inches tall. And it's, as far as I can tell, it's completely eliminated. And here we didn't screen it against other grasses, but you can see you definitely have a very diverse canopy there. So I'll wrap up by just kind of trying to, hopefully I'm reinforcing some of what Luke said. When you're trying to be selective, just let's say qualitatively ranking them, those grass herbicides are going to be your most selective approach. That selective pre-emergence is going to be, it's still going to be selective, but less so. And then kind of bringing up the rear from the selective approach is when you're doing that selective pre-emergent material plus that low rate tank mix partner. And with that, I'll stop and happy to address any questions.
Great job. Thank you both so much, Art and Luke. I had a few quick things. I mean, I think we should address the fact that Luke didn't feel that glyphosate was really that useful, and Art does use it, so we should address that. But I also had a question about, I don't think either of you has looked into vinegar. That didn't come up. There's the high concentrated vinegar products. I've been kind of curious about those and trying them out, in certain places. And then years ago, I remember when I worked for Park Service, a guy over at Huntley Meadows was testing very low rates of Roundup glyphosate at the time. I think it was rodeo because it was in a wetland setting. He was using like 1% glyphosate early in the growth season of Microstegium and having good results where the natives would show up. You may have remembered that. And then I'm also curious about what about in stream valley settings where you have just a massive amount of stiltgrass that's kind of taken over a restored stream valley area.
Well, so the vinegar, I mean, here's the thing. Vinegar is largely non-selective. I'm going to guess, if you apply enough vinegar to burn and vinegar is literally just a burn down product. Herbicides that we call burn down products still actually have some systemic activity. They just work so quickly that they don't get a chance to translocate through the entire plant. Vinegar is just burning the plant. So I guess it boils to this. If you're working in it, so you as a contractor, if someone says, I want you to use, OMRI certified materials, you're kind of looking at vinegar or fire, maybe some of these other oils, but it really is just burning it back. It's burning everything back. It's chemical mowing. So, I think you just have to decide, am I really gaining anything here? Because they've set everything back.
And it's acidic. That's the other thing. A lot of us are concerned about if it was used a lot in an area, it can increase the acidity, decrease the pH of the soil. But people want to use it because it's kind of an environmentally friendly product, and maybe it's useful in some places.
Well, that, I mean, that's, of course, that's like a whole month of discussion there. Is it
Yeah,
environmentally
sorry.
friendly? I mean, because so it's questionable whether it even is. The other
Mhm.
thing is it's so much more hazardous to the user
Yes.
than any of these selective, synthetic materials. And, pretty much, I think I don't know, but I would suspect, and maybe somebody can verify, but, anything that you're contacting with the vinegar, I should point out, herbicide type vinegar is a 30% concentration of acetic acid, maybe 20 to 30. Household vinegar is 5%. So you're using this much more concentrated material. And I have to believe anything you're putting it on is not going to be happy, regardless of the taxa,
Mhm.
arthropod, plant, little furry creatures, they might not mind it. Well, certainly if I was an amphibian, I don't think I'd want any vinegar on me. So
Yeah,
that's,
I mean, if that's a question that it probably comes up a lot, and this is not my area of expertise, but my comment on that is there needs to be it's like a general education on herbicides. People want to use organic, insecticides. Some organic insecticides are like some of the most toxic things. I think that's a whole other discussion. And there's a question on the chat about, I think it was about grass-specific herbicides effects on invertebrates. I think they're minimal to none. There's maybe some, minimal to none. I will come back to the comment that you said about glyphosate. So Art described much more, and again, the caveat that I'm not a weed scientist. I'm aligned with weed scientists in my group, but I am not an herbicide person. I am not a weed scientist at all. I'm a community ecologist primarily. So Art was describing a lot more complex vegetation management situations than I was. If you're trying to manage multiple species at once, if you're trying to manage for timber and low understory vegetation density, much more complicated situations. I was talking about on a small scale per plot situation where you're trying to keep some native herbaceous species and tree regeneration. Very specific to the situation that I was focused on in my research. There was one other comment on here I wanted to respond to, which was about the cleistogamous versus chasmogamous seed production. These are the seeds that are produced, the flowers that are produced at the end of the stems that most people recognize. Those are the ones that can possibly outcross versus cleistogamous seeds, flowers that are in the within a leaf sheath low down on the plant. And that's why you need to mow very close to the ground, which is a problem because then you're obviously causing more damage to native species that you probably want to save. There's another question about mowing and repeated mowing and exhausting the seed bank. Depends on where you are. It can take years to remove all of the stiltgrass. But even in 1 year, in a lot of cases, you can, if you time it right, like Art was saying, and mow before seed is ripe, which would be by the end of September in most areas, then you can reduce seed production and reduce the size of the population the following year to some extent.
I have a quick question about the cleistogamous flowers and seeds. When do they get released?
Well, they don't really get released.
Okay.
So I've seen seedlings, rows of seedlings, because a stem, floated away or something. Right. And then as the stem decomposed, they emerged out of the stem. So you have like a little row of seedlings. That's one of the issues with moving, like the picture Art showed of the litter or the thatch stuck in the in any kind of equipment, is you can be transporting seed that way.
Mm-hmm. Okay, thanks.
In terms of the vinegar, I want to emphasize what you touched on, which is vinegar is actually, especially the horticultural, more concentrated vinegar, it's actually quite dangerous. There are higher requirements for PPE, that is personal protective equipment, with vinegar than with many other herbicides. It can cause blindness. It can burn your skin. Vinegar, people think, oh, it's natural, it's safe. This vinegar is not a safe product to use. Something like glyphosate, which a lot of people are afraid of, is a whole lot safer.
Brandon, you can unmute.
This is kind of like a broader question, but it definitely applies to stiltgrass. You know, we talked about the soil disturbance that happens with hand pulling. Is there potentially, from a management perspective, an opportunity to pair that soil disturbance with seeding, like desirable seeding at the same time? Could that be kind of like two steps in one of a positive step forward on stiltgrass?
So I'll say yes. Some of the initial work I was doing was with Dennis Burton. Some of you might remember from the older days of the Mid-Atlantic Exotic Pest Plant Council, back when it was that, in Philly. And we looked at, now we were using glyphosate late summer to get rid of the stiltgrass, and then seeding a native cool season grass mixture, a lot of Alimus species. But that scenario you described would provide that same opportunity. Come in August, early September, take that stiltgrass to the ground. You're going to get some soil disturbance there to facilitate that seed to soil contact. So I think that would be definitely a means to do that.
I will say with hand weeding that our plots were 2 by 2 meters, and sometimes those 4 square meters might take, I don't know, 45 minutes or an hour if you're trying to be careful. And I often hear about stiltgrass invasions described in acres. And so it's not very practical for extensive, and it's not at all practical for extensive invasions.
Jill, you had mentioned someone saying they were using low rates of glyphosate early in the season and seeing good results. I think it's going to depend on, to a certain extent, the spectrum of species that are there. I think the early season has the advantage of, particularly with perennials that are there, is your net carbohydrate movement is, towards the tips. And when glyphosate becomes really problematic, even at sublethal doses, is later in the season as it starts getting translocated into the storage tissue. So that early season approach might be a wrinkle that gives you a little more selectivity with the glyphosate. The images I showed, the glyphosate applied in July, even at really, really low rates, was pretty devastating.
Okay, thanks. Okay, it looks like Peggy Funk has a question. You want to go ahead?
Yes, thank you. So I'm noticing at the end of stiltgrass season, like right around now, as you look sort of into the woods, you'll see, all of the dead stiltgrass. And if you go out and start working in that, it really seems to be matted. And I'm wondering, is it helpful to rake up all of that matting to allow new growth to come up, or does it not even matter? Say you wanted to replace it with some other plants or some seeds or something, but, it's really matted. It seems like it suppresses all of the potential natives that could be out there.
Well, it suppresses the Microstegium as well. So if you remove that litter layer, the seed has already fallen from the chasmogamous seed production, and there may be some in the litter, but following litter removal, the emergence of stiltgrass is phenomenal. I had one land manager that I worked with sent me an email, this is years ago, and said he'd counted over 150 seedlings in a square inch. Because of course, all those are not going to grow to adulthood. There's incredible density dependent growth, meaning that even a few individuals will grow larger if they have more space, if there's lower density. So this is where there was a question about fire, and I did a lot of work with fire during my postdoc work. and most of it was work on the interaction between the invasion and fire and then how it affected trees. But we also looked at the success of Microstegium if you burned it during the dormant season. And if you did that the following year, there was about 300% more Microstegium biomass, because some of the seed dies, but it produces far more seed than it needs in order to sustain a successful population. The only time, and we have one paper on this that was kind of a odd study that I did with collaborators, is to use a propane torch to burn Microstegium during the growing season. It's a bit silly because you can kill individual plants just like you could with a string trimmer or a mower, but of course, fire's not selective. You can get resprouts of woody species, I suppose, but yeah, stimulation of seedlings to emerge by removing the leaf litter is a huge issue.
That's very helpful. Thank you.
We have a question in the chat from Ralph. How many years/seasons is the seed bank viable?
Yeah, I don't think it's been totally sorted out. It, just like with a lot of species, it depends on where we're talking about in terms of everything from latitude to soil type to soil moisture to soil microbial community, but it's at least years. I'm guessing something like 3 to 5, maybe 6 years, probably, or 2 to 4. Not a long time. It's not real hard seeded like garlic mustard. Garlic mustard can last a very, very long time.
So I will share an anecdote that I'm probably not supposed to, so I won't identify the people, but colleagues of mine, probably starting around 2011, were engaged in I'll say a stiltgrass elimination study where they would look at different suppression techniques every year, and I don't know how many times they repeated it, but the next spring, or they would take soil samples, cores, grow them out in the greenhouse.
Mm-hmm.
And they said for at least the first 6 years, they didn't see any reduction in density of stiltgrass seedlings coming up in their cores. I don't know where they are with that now, and I kind of have to get after them to publish that, because I think when they finally do, it's going to be very interesting and maybe slightly discouraging.
Yeah, that's the way to do it. That's how it's been done with garlic mustard.
Art, can you talk more about, you talked about planting trees. How successful is it to plant trees and shrubs and try to keep them going and keep weeds away from where the roots are to suppress stiltgrass? I mean, it is a shade tolerant plant though, so it's a little tricky.
Yeah, well, and so 2 points there, starting with your last one. When I recommend reforestation to people, because I think it's easier, I also point out that keep in mind, you're releasing that next generation of woody plants, that next generation of the woody community, but it's still going to have stiltgrass in the understory. In our part of the world where deer are so prevalent, one of the things we're challenged with is, developing these park-like situations where stiltgrass becomes the lawn and you have these mature second growth trees and, no younger generation. So I really do think operationally it's easier to plant trees, put them in tubes, maintain them in those tubes for, and there's different schools of thought on how long you keep trees in shelters. For instance, Stroud Water Research Institute says just leave them on to prevent buck rub. But anyway, point being, if you think that really the problem here is the deer, suppress the stiltgrass, get faster growth, get them out of the shelters, get them above that 4.5 feet. And at that point, you could take the shelters off and just kind of say, okay, next generation woodies released. And they're going to provide that ecological function that you want, and they're definitely going to have stiltgrass in the understory.
It's complicated.
I used to joke, and maybe it wasn't really a joke, that, one of the elements of integrated pest management was, we'd say cultural, mechanical, biological, chemical, and attitude adjustment. In dealing with these seed banked annuals like stiltgrass, like mile a minute. I'm assuming the arthraxon is gonna be the kind of the new thing behaving similarly. Is you just, rather than trying to get rid of them, it's like, let's just figure out what desirable plant communities we can release that are still functional in the presence of these invasive species.
I will say on that point, so we have this paper on our long-term plots where after a few years, the trees, we crossed a fire treatment in these plots. And when we added fire, it killed the emergent trees and the perennials became dominant. When we didn't add fires, the trees became dominant. And in both cases, stiltgrass was almost entirely eliminated. The huge caveat in that paper is that this was a bottomland forest area with incredible amounts of resources, high soil nutrients, high soil moisture. It was very, very productive. And so we point out in that paper, and we used to point out when I would talk about that study, that in a lot of places, stiltgrass has been around for decades. So in some cases, if you get native species to establish, like what Art's talking about, they will become dominant over top of stiltgrass, even though it's very shade tolerant.
Okay, thanks, Luke. Dee Davis, you're on.
I volunteer at a park where we're not allowed to use any herbicides any longer because this PFAS controversy, that there's PFAS in the herbicides, and I don't know if it's in the herbicide or in the plastic jugs it's in, but anyhow, and I can understand why they don't want to take the chance right now. And of course, we have stiltgrass. So we're trying to get rid of it. One of the ideas we had was to throw a silage tarp over it, kind of like how the organic vegetable growers use silage tarps to encourage the stiltgrass seeds to germinate and then fry them because they're going to be under this black heat. And I didn't know if there was someone else who has tried that and if they think it might make sense, or it just sounds stupid. we don't know what else to do. So we have an area that was nothing but stiltgrass, and unfortunately we just took away all the biomass off of it, and then we were going to lay this tarp on it to cook it essentially for a couple months, and then seed it with something desirable, the grasses and native herbaceous perennials.
I suppose that if you left the tarp on long enough, it would cook the soil, kind of like if you're solarizing your garden.
I mean, I'll emphasize, if you wanted to do that approach, I would do exactly the way Luke described it, where you use a clear plastic, because the soil is going to get much hotter. Because what the clear plastic's doing is allowing all the energy to strike the soil. Rather than heating the plastic, you're heating the soil, and you're actually trapping a lot of that heat under the plastic. What I don't have any sense of is how deep you can cook the seeds in the soil. I almost feel like the pragmatic approach there is, well, there's a couple ways, but I really do think reforestation, establishing woody plants is generally going to be an easier approach, where you're kind of limited on the tools at your disposal, because really then you just need discrete areas of suppression to establish your desired plants. If you really want to go with the seeding, I would definitely play around with the solarization and see if you really can cook it. I would also think too about just keep it from going to seed in 24. And then, as you get into August, mow that down, and put in I would go with a native cool season grass dominated seed mix that will get established in the fall. So then at least next year you will have a native stand that's going to have some stiltgrass in it. Or the other approach is, I guess you just take a few years and see, can we really cook the soil enough to reduce that seed bank pressure from the stiltgrass? I don't have any kind of quantitative sense of how effectively you can cook soil with solarization.
Okay, thank you. Okay, Judy, go for it.
I believe that the solarization extends about 2 to 4 inches under the soil surface. You are going to get, of course, other seeds. So if there's a seed bank of native plants, you're also going to be solarizing those if they're in that thin layer. In addition, you can affect the beneficial microbes in the soil with solarization. So that's just another couple of things to consider, and it may be worth it to get rid of the stiltgrass. It may be worth it to damage the soil a little bit if you're successful in the end.
This is just the tip of the iceberg. I feel like we've all put on our ice skates and we've skated out in the middle of the pond and it's starting to thaw and we're like, because there's no easy answers with this plant. But we did get a lot of really good information and a lot of, stimulating ideas for maybe what we might do. And really appreciate Art and Luke for speaking to us tonight. And who knows, maybe we'll get in touch with you again for some follow-up questions. But thank you both very much for presenting. It's a really interesting plant, and you got to give it credit for being so persistent and so difficult. But we kind of all wish it would get a disease and go away. Well, thank you all very much, and have a good evening. Enjoy your spring. Don't get too stressed out about the invasives, but do what you can. All right. Good night, everyone. Thank you.